publication, I chose to focus on the Philpott Lab, which is a lab that started at the University of Chapel Hill and focuses on neurodevelopmental disorders. The publication specifically I chose to focus on is the molecular and behavioral consequences of UBE3A gene overdosage in mice. This lab was made, and the main researcher at the lab is Dr. Ben Philpott. The particular publication on the molecular and behavioral consequences was also written by him. The gene known for causing the neurodevelopmental disorder DUP15Q syndrome is what this research focuses on, and Dr. Ben Philpott is the principal investigator, and he also works at the Office of Graduate Education at UNC Chapel Hill. This research covers neurodevelopmental disorders. These disorders have impacts on the brain and the nervous system, which are the main control systems of our body. Specifically, the DUP15Q syndrome covered in this paper affects people intellectually, impairs their motor abilities, which is their movement and coordination, and can also result in autism spectrum disorder, which is a main developmental disorder that impacts millions of people in America. Researchers hypothesized that a specific gene in the duplication or the overexpression was the main causation of this disorder. This gene that most commonly passes on from mothers to their children is called the UBE3A gene. Scientists wanted to place this gene into mice to test the effects extensive amounts of this gene can have on the mice's brain and their nervous systems. Mice were known to duplicate humans very closely, so mice were the best organisms to research. Researchers first needed to find a way to assess how the overexpression of this gene that causes this neurodevelopmental disorder affects mice. This protein, UBE3A, needed to be generated into the mice. Researchers looked at how mice's coordination, anxiety levels, and memory levels differed throughout the experiment. They used the Morris-Water maze task. This task consisted of a 7-day training phase. Mice of all different genotypes, meaning color, size, and gene makeup, were tested. The purpose of this task was to see how mice utilize their spatial cues and visual cues to locate a hidden escape platform that scientists made within the maze. During day 6 of the trial, after letting the mouse find their way through the maze to the platform on their own, researchers removed the platform from the maze in order to gauge the mouse's memory for the platform's former location. This way, they were able to see if the UBE3A gene impacted mice's memory in a certain way. Results of this Morris-Water maze experiment where researchers were able to see through a heat map-based visualization, which is a visual graph that uses color gradients to represent the density of certain data points, which in this case the data points were mice, in a certain area. For example, in certain areas the color would be darker or deeper if the amount of mice in that area was higher. The number of platform identifications in the target quadrant of the maze was similar across all genotypes of mice, meaning that the number of mice that made it to the target quadrant in the maze was similar in all different types of mice, showing no differentiation depending on different genes. Researchers were able to conclude from this information that associative learning and memory, and spatial memory, was unaffected by the overexpression of the neurodevelopmental disorder causing gene, UBE3A. Importance of these findings and research. Researchers were able to understand from this particular portion of their experiment that the presence of the UBE3A gene alone is not responsible for the neurodevelopmental disorder DUP15Q syndrome. Though, figuring out this information is crucial for the development of research like this. They can focus on other genes or disease-causing agents that contribute to the neurodevelopmental disorder. While UBE3A was found to be a critical part in the development of the disorder, other genes cooperate with it to produce the disorder.